Hola Colombia, hola Bogotá. Muchas gracias por su invitación. Janet y Andrea. I cannot speak good Spanish unless I am drunk. So, your job, if you want to communicate with me, is give me drinks. I am so excited to be invited here because I believe in the ICGEB, which funds research in South Africa, my country. And I believe in what the ICGEB is doing here. And I believe in the science very much. The quality that I have been exposed to over the last 2 days has been superb. And so I'm very excited to be part of your world and our research together. So today we're going to talk about translation. And this is a complicated concept. Let's start with what happens if translation is effective. Doctor, I have a cold. And the doctor, his response is, I know. If you think about what that means, if the doctor knows your problem before you come to see him, or you don't even have to see him, that is the aim of effective genome medicine. So let's discuss this concept. Medical care is educated guess. And by that, we know when you present to a doctor, the doctor refers to previous knowledge about your condition. What has happened before, what might be your problem. It's a guess until a correct diagnosis. But if we can learn more about what has happened about to people like you, then the doctor's job is much easier because they can utilize information that has been gathered previously and systematized. So let's think about the concept of translational medicine. It's the combination of something I think we all love, scientific discovery, and its translation into the improvement in health. Perhaps if we do scientific discovery in biomedicine, we think we are contributing to health, but we're not. Because without translating our discoveries, there is no real impact at all. It's just a good paper and recognition by your peers. Donald Trump likes that, but he's not very effective so far in changing the world. So we move from the biological, our understanding of the molecular mechanisms, to the clinical, where we interpret these molecular mechanisms in the concept of systems of disease, symptoms, diagnosis, and drugs. So let's think about this concept of your genome will be your guide. Imagine if, as part of the registration for this conference, you gave temporary permission through your app for your genome or part of it to be shared so that the interpretation engine that was part of the conference told you that there were 3 people in the audience with you who shared your interest in cilantro or liked to tell you that you have a common ancestry or common job. These are very interesting concepts, but they don't really have anything to do with your health. And if you think about that, we are unable to exploit the genome without a technology to exploit the genome. Van Leeuwenhoek was the first person to see a living cell. And that was through the utilization of the microscope. It became possible to realize that one of the units of biology was the cell. Until prior, that was not known. And the microscope opened up an entire new version of understanding of human and health and the world. Now we have technologies that are machine-based that read the genome, next generation sequencing technology. And this technology has one basic facet, and that is it can generate genome sequence information faster and faster. It's the same concept as the Model T Ford, which, if you think about automobiles prior to the introduction of the Model T, they were very, very expensive. The Model T reduced the cost of ownership 10 times. It was still expensive, but it was cheap enough that 15 million of those units were sold. So the concept of an enlightening technology like next-generation sequencing means that we have the potential to action genomes in the context of medicine. So one of the concepts of that is customized medical care. We know you won't respond to this drug, so we won't give it to you. Or we have a very cheap drug we can give you that will help you a great deal. And it won't help your mother, but it will help you. So in order to do that, we need to understand the genome that is you. And frankly, we don't know very much about it at all, as you know. Potentially, we need to predict your response. And there are a great number of capture systems to predict drug response now in genomics, and I'll explain a little bit of that later. And perhaps we treat you as a well person. We treat wellness instead of illness to prevent you from becoming ill, because we know what beneficial aspects of your genome we can make stronger, ward off your infectious disease. And of course, as I mentioned previously, if you know your genome and someone else knows their genome and you can participate because of common aspects of your genome, you can participate in a network of people who have the same illness as you. You can join together to support each other. So that's all very well. Those are the concepts, but the reality is quite different, which is why I use the word reality in the title of this talk. There is this concept of genetic testing, which is performed worldwide. Genetic testing is common everywhere. And aspects of your genome can be revealed from that today. New treatment guidelines are in generation now based on genome information. A very simple example of that is whether or not you will respond positively or well to treatment by the blood thinner warfarin. Some people cannot take warfarin, and that is a genetic condition that can be described. And of course, prediction of how to better treat a patient is the goal. And financially, that's important when you have single-payer systems that really want to save money and be more efficient with a limited cost. I thought I'd share my genome with you. I got a packet in the mail from 23andMe, and it said, for only $99, we can tell you about things which you really don't want to know. And I discovered the following things at some level of risk. Now remember that the risk system that is being used is a chip. It is sampling information very selectively from my genome. It's not likely to be that accurate. So clinically, this prediction is not really very strong. But let's look at the anecdotal information. I don't yet have prostate cancer. I've had psoriasis since I was 21. I haven't had gallstones, but I've had kidney stones. My father died of colorectal cancer, and I'm waiting for this one to come along in a little while. But there's one that I was very excited about because it was absolutely true. And that was... so that was great. And I was able to tell my sons, who also took the test, you don't worry, you won't lose your hair. So this is very effective utilization of genome medicine, but it's anecdotal and it's not very clinically robust. Let's think about population-level sequencing of the human genome. Today, there are projects occurring in the following countries. The smallest one is in the Faroe Islands, 50,000 people being sequenced. The largest active project right now is for 300,000 genome sequences in Qatar, where they are attempting to sequence the whole population of Qatari citizens. In the United Kingdom, 100,000 sequences are being generated. In the United States, they intend to find 1 million cancer patients. And in Brazil, there's a project as well. So I think this is very exciting. The problem is, what do you do with that information as and when you generate it? Well, let's think about this concept of technology helping us to find therapeutics. We start in the 50s, the concept of biochemistry and pharmacology. Groundbreaking ideas in rational drug design. Then we move on to affinity assays, ways that we can interpret outputs of our genome as product. Then in the 80s, when I was a graduate student, the big technology to get into was monoclonal antibodies. something which could identify and target very specifically within the body. And now we have drug budgets, huge budgets to be spent on acquiring data, most of which is related to the patients and their phenotypes, but some of which is being invested in understanding the genomes of patients. So genomics together with phenotype information are actually the new assay technology that is likely to change our interpretation of the ability to develop drugs. So let's go through that concept in this slide. There's first of all the first human genome project, many millions of dollars to generate one human genome. And then as we crest the wave, we have many whole genome sequencing projects occurring simultaneously. And through those, we're trying to interpret that information through genome-wide association of phenotypes together with the variation we see in the human genome. Projects such as ENCODE, looking at the functional differences and commonalities within genomes. And we get to this concept of the quantified patient. I have this number of blood cells in my blood per unit time, per unit volume. And that can give you an indication of my wellness today. All of this is data. And data needs to be stored and manufactured. And as you heard earlier, the influence of companies such as Microsoft, HP, Google is very powerful. And then we have the buzzwords, giving us the volcanoes and the mountains much, which you hear, the big data concept, which I will not use that phrase again because it has no meaning. So one of my influences is Lee Hood. Lee Hood was responsible for the marketing, development, and rollout of the automated sequencer, which was the precursor to next-generation sequencing technology. But he said something very important in 1993, and that was during a SmithKline hiring spree at a company, at a meeting. He said, discovery is becoming a data science. He underestimated it at that time, and he said at least 25% of researchers will be data researchers looking at data. As you know now, that number is changing very dramatically in terms of biomedical research. Another aspect that is changing our world, worldwide, it's happening everywhere, is that clinicians are learning to type instead of write. They are putting information into computational systems which can then be assessed and put together in clinical health records that become electronic. In UK, this is a pretty primitive story. I am a recipient of UK medical health. I go to the general practitioner. He writes something down on a piece of paper. And his database maybe is a Microsoft one on his computer. It has no link to the hospital. That's the state of the art in UK. In countries such as South Africa, where we've not had a legacy of good medical care, new systems are being put in place that are very efficient. So there's a disparity in health that is very much exposed by access to electronic health records. So let's go to this concept of knowing something about the population. This is a common mutation that I have, that all English-derived people have. And this is a common mutation that Peruvians, in particular Amerindian, will have. This information is useful because that common mutation can be a mutation in ApoE. The gene that if you have a mutation in one particular position will give you a predisposition at a level of as high as 40% to getting Alzheimer's disease, unless that common mutation might be from a population that you're from where it is protected. So your population is very important information. I explored a little bit of that. There is one paper on genome medicine in Colombia, and it's by this group, Computational and Restrepo, and they found a history of genetic information in Amerindians dating back to 600 BC to the Moquillo syndrome. And as you can see, it's a dysmorphology. So there's been, due to consanguinity and due to the inbreeding of certain populations in certain families, there's been a prevalence of certain genetic diseases that are caused by a single gene. The application today in Colombia of genome medicine is very much in the area of care to underserved populations, newborn screening, and an increase in the number of non-clinician geneticist professionals, some of whom may be you in the future or today. the real application of genome medicine today is in diagnostics. And the Americans waste no time in thinking about generating companies that can help us understand ourselves. 23andMe is an example, and I showed you what they did. But really, these companies are not performing diagnostics. What they're performing is data capture about you. And if you think about it, a company such as Illumina Helix will happily take your saliva, generate within 1 week a genetic profile, and suggest things that you should do to remain well through an app. But that app ecosystem relies on your genetic profile in a database. And it's the health insurer who's interested in that information because they can provide much more specific information and much more specific identification of the risk of insuring you. That, of course, has major implications in terms of social wellness. So Craig, who wastes no time in having his own genome sequenced and contributing it to the human genome, has started a company which will, for $250 at Discovery Health in South Africa and in UK, look at your exome, those coding regions of your genome, and take that information from you, and we'll sell it again with your permission. Most people don't realize it's being resold, and his company is not a diagnostics company, it's actually a database company on you with this highly detailed information about you and your genome. It's a very effective way of understanding which population might be sensibly responding to a particular drug, and you can sell that information. In fact, a company he's generated called HLI that gives you a complete omics workflow. For only $25,000, you can get a virtual reality headset for free, and you can wander around your own genome and your own quantified health after it's been examined by a group of experts who will give you constant updates on your quantified information about yourself. But the concept is a data concept. The company is the database. So let's think about the genomes that we have. Each of our genomes is quite variable. In fact, if we look at any two individuals, there will be as many as 500 million different human variants between two individuals. And even if they're genetically identical, you'll see variations. And brothers and sisters, of course, you'll see enough variation, you'll see less. So the thing I want to point out is that understanding this variation in terms of disease, do you have an A, a C, a T, or a G at a specific position in a specific location in a genome, say in your ApoE4 gene, can give you indication of disease causing. If you present with a known disease and it's a monogenic disease, but it has not yet been identified, the clinicians and the scientists will pore over your genome sequence, will look at the medications that you're taking, will look at the publications, and will come to build evidence that a certain gene in your genome, a certain location, may be contributing or giving you that particular genetic disease. These are called pathogenic variants. And to be absolutely sure a variant is pathogenic is very difficult, but it is possible in some circumstances. And it requires supporting publications and an understanding of guidelines. We teach this as a discipline. A whole master's degree is dedicated to this particular concept in the UK because the 100,000 Genome Project there is seeking to build a corpus of knowledge around understanding genomes so that nurses and clinicians can work together with your genome to understand you and give you advice or to give you treatment for pathogenic variants. So an interesting paper on this is by Dan MacArthur in Nature. It's great. It gives you a really good background to this area. So this is the workflow of that kind of concept. There is a genome medicine center that will be set up or is set up. And basically they recruit patients who have rare diseases or cancers. And they do a clinical next generation sequence lab validation. They do a test kit. They look at your raw sequence data. They bring in images and data. And they put this into something called a smart city health arc. What that means is money to bring people together into an arc that in some way will help us understand the genome better. It's very nebulous, but we're trying. And these clinical data centers develop new methods and machine learning to in some way then provide a clinical genomic outcome. This is the concept. It's being built right now in UK. I think it will take quite some time. But the idea is you move from a next generation sequencing understanding of the genome to a clinical report that has meaning. So this variant interpretation game is a game. And it's again because we really don't understand the genome very well at all. We know a lot more about the galaxy than we know about the genome. We've been studying the galaxies for a lot longer. So genome-wide association means variants which simply are found more frequently in people who have a specific phenotype. They're tall, they're bald, whatever it is. And that genome-wide signal often does not occur in the coding region of the genome. It just occurs somewhere in the genome that's non-coding. And as you know, there are two alleles, but you can have any two alleles because we have two sets of genomes within our body. So you have different genes from the same pair of genomes being expressed in different tissues. So this is called allele-specific expression. The genome is not laying out in a single layer. It's folded, so interactions occur between regions of the genome that bind to each other. And this functional interaction occurs after the transcription of a gene and its translation into a protein. So really we're thinking about an orchestra of regulatory elements, variants, and critical positions which define what kind of music you're going to play. This is a quote by Alban Sandlin, who's a fantastic gene regulation expert at Karolinska. Also, you have these single nucleotide polymorphisms, which are variants, which are linked to specific expression traits, which we call them expression quantitative trait loci. We use this in potatoes as much as we use in humans. And all of that then allows us to think about people who have one sort of response or another sort of response due to genetic stratification. So this is a complex slide, but what I'm trying to tell you is that interpreting the variants in the human genome is non-trivial. So Terry Pratchett is an author that died recently in the United Kingdom. It was a great loss to the country. And he was diagnosed as potentially of having had a minor stroke 10 years ago. What he had actually was a mutation in his ABCA7 gene, and that mutation was a misdiagnosis, was not diagnosed, and as a result, they didn't know he had an early form of dementia and Alzheimer's disease. What we are doing, together with Rudy Tansey's group at Harvard Med School, is interpreting the sequence of 1,500 Alzheimer's genomes in families, 437 families, and because of that, we've discovered new variants that may cause Alzheimer's disease. Now, that is not clinically interpretable yet, but that gives you an indication of what can be done with just 1,500 genomes at a cost of $6 million at the time. It's much cheaper to do that now, but it gives you the concept that we could do something if we knew more. Another famous scientist, Stephen Hawking, has motor neuron disease. He has a mutation that nobody knows about. They don't know which one he has. No one's actually looked at his genome, but it's progressive, and that disease is made up of a handful of known mutations and many unknown mutations. And we study it at the institute I work at. It's also known as ALS, amyotrophic lateral sclerosis, or Lou Gehrig's disease. The point is that if we knew the mutations that were causing this particular disease in detail, and we knew more about its function, we could intervene potentially to slow the progression of the disease. And that's something that excites me, because that brings us from thinking about clinical medicine to biomedicine to translation, and going back then to a new treatment which we can trial and change the course of diagnostics. So there are 15,000 samples now being sequenced for ALS across Europe in a project called Project Mine, which we're part of. About 5,000 have already been sequenced, and the first paper's come out discovering one new gene that contributes to that disease. And effectively, that's a crowdsourced operation. If you know anyone that has motor neuron disease and you care, you can have that person's genome submitted, you can contribute some money of your own, and eventually you may learn more about the disease. So let's think then about therapy. Well, it's very easy to treat an animal. And most of what we're doing in terms of treatment is curing animals of disease. And that means that we're good at veterinary therapy, but we're not very good at human therapy. So what we need to do is utilize models of humans or humans themselves. And the move right now is to perform research on humans themselves in small preclinical trials by volunteers who are ethically approved, and to take the cells from patients who are sick, their fibroblasts, their skin cells, and to transdifferentiate those. I was really excited to be picked up on my first day here by a student who's performing transdifferentiation in Parkinson's. And it's a technology that's very powerful. It's not to be estimated. Some colleagues of mine, Kevin Egan and Clifford Wolf, have generated the first transdifferentiated cell model for a disease in ALS, and they rescued that ALS phenotype by taking fibroblasts, turning them into motor neurons, and then treating those motor neurons with a partially effective drug to show that the motor neurons worked a little better when they were treated. So that was a pilot that's now being expanded to clinical trials. And what that means is that 200 different compounds are passed across transdifferentiated motor neurons so that they can, the company, SmithKline, can test which of the compounds the patients are responding to best. This is cool stuff. Both of these projects here I'm mentioning, I won't mention this one in detail, but require the full genome sequencing of these transdifferentiated cells because we want to know, are the genomes intact? So now we get to stuff which is the bread and butter of bioinformatics, and that is the concept that genes don't hang out in the desert by themselves. They go to the pub, they go to the bar, and they get drunk with their friends. People like to talk to each other. Genes like to talk to each other. Gene products like to interact. Genes do not work alone. If we understand that as a basic principle, then we understand that drugs don't really target single genes very often. They often target sets of genes, and we're never quite sure which sets. So we better understand that concept in networks. So what you can do is build an integrative model which progresses. You can start by saying, okay, let's take information from human cohorts, from fly and mice, and look at their genes and find out which of their genes interact in a mouse model or a fly model, and then also in the cells of patients or in their blood, and ask the question. If I looked at all those interacting genes by virtue of the fact they all express in similar ways, they co-express, can I reduce the number of genes I look at and move towards some subset? You can, by introducing new information that allows you to quantify the importance of individual genes and then to ask about their neighborhood. Are they in a good neighborhood? Or are they the bad guys wearing leather jackets and driving Harleys and hanging out somewhere pretty nasty? Well, maybe that's where the nasty place is. Let's take some more interest in that particular aspect, in that sub-module within the network. So this kind of concept has been put into practice. And a colleague, Eric Schatt, has written a great paper in Cell on discovery of a set of genes which appear to modulate the response of, modulate the progression of Alzheimer's. By assaying hundreds of post-mortem brains and looking at the gene expression in those brains, they were able to build a submodule of expressed genes in the same methodology I've just showed you, and to find that this gene, TYROBP, which is expressed incidentally in glial cells, which, as you know, have a role in the immune response in the brain. And he found that gene as being very important. It's a key driver. Well, the community has not jumped up and down and got that excited because there are no mutations which appear in that gene with patients who have Alzheimer's. And people want to see mutations because the concept is in our heads: mutation, illness, pathogenic. But this is a truer representation of what's going on. It's being influenced by existing mutations. And that gene, when it's dysregulated, allows us to understand Alzheimer's. So, taking this concept forward, we've become part of a consortium called the Accelerated Medicines Partnership. And this is a network sharing consortium. And what we do is that we build networks from sets of these post-mortem brains in gene expression, in protein-protein interaction, in proteomics, in metabolomics, and then we try and build some form of understanding in terms of a network together with companies. And an example of a company is Takeda. And they've said, we will validate your model openly, which means that if you say this subnetwork is important, they'll go ahead and test it in patients and in models and use all their drugs and tell you the response they've got openly. And the reason for that openness is, of course, they get an understanding of the system very quickly, and at the same time, that information can be shared with the community for better understanding of the mechanism of Alzheimer's disease. So this has an ethical strength, but in reality, what it means is we have to do stuff, and we have to work with omic data sets. These kinds of data sets are an example of what we work with, mouse models, and the network is the data, not the gene. And what happens is that sets of subnetworks become available, and we generate a target leaderboard. Each group says, I think this is the most important set of genes. I think this and this is one. But because we're academics, no one shares the names of the genes. So what's the point? And that's the question that's being asked right now in this consortium. I find it very fascinating. So I've got about another 10 minutes to go. What time is it? Okay, we're doing great. Okay. So I want to tell you a little bit about bioinformatics and its role in this problem. Some of you are not bioinformaticians. I've met many nutritionists today, all kinds of cool stuff. So I'm going to gloss over some of the algorithmics. But what I want to tell you about is pathway fingerprinting. This is a concept that works reasonably well in our lab. What it means is that we look at gene expression and we say, we know that there are pathways. Some are active and some are not. And some are more active than others in patients. The question we ask is if we take highly expressed and lowly expressed genes across a sample, we put them into a pathway, and then we ask how active are those genes in that sample, according to their rank, in a gene expression system, we can then find statistically a distribution of response, and we can just label it into the French flag. We can say some are highly active in this extreme, some are not active, and our pathways, our single pathway has a different range of activity. What we can then do is just do this again and again for many, many, many pathways, and we can build what we call a vector. It looks like a fingerprint. We can do it in humans and mouse, and that allows us to put ones and zeros instead of lots and lots and lots of genes, which allows us to compute upon this information in a simple way. We can scale this hugely across thousands and thousands of people, thousands and thousands of experiments, and that allows us to quantify our patients and to quantify the system. That in turn means with the fingerprint, we can find fingerprints which are diseased and fingerprints which are normal. That in turn means we can come up with an assay for a patient that walks into the door and say, as I said to you before, some patients are going to group with the not so well group. This is the treatment they'll need. Some patients are going to group with the well group. Distance from healthy. And that concept is an exciting one that we're doing together with a company I'm working with, Biogen, in multiple sclerosis, and a group we're working with in the UK, where we're looking at blood that's taken from MS patients. And we've already discovered that you can predict how rapidly a patient is going to progress based upon this methodology. We can also predict survival. We can say in a wound healing model whether you're going to do well or not. And effectively, this is the part I think is very sexy. We can incorporate genetic variation that is yours into this model by plugging in genes which have showing association with genetic variation into the gene expression system. We can combine this with genome-wide association studies, sampling lots and lots of blood with microarrays and RNA-seq, with disease signatures, and with response to drug signatures in one integrated fingerprint. And the result is we can find out how all of these networks in the system interact with each other. Now that sounds complicated. It is complicated. I'm going way ahead of genome medicine here, but actually this is not rocket science. This is simply asking a very basic question. If I take this set of genes from me, and I take this set of genes that are known to show a response to aspirin, and I ask the question, do these gene sets talk to each other? I can ask how much they talk to each other, and I can say, the headache I have right now, although I didn't drink last night, which is a rare event, it's too beautiful to drink here. Will I respond well to aspirin? And if you think about environmental response, will I respond well to aspirin today? Okay, so we're trying to quantitate this problem. So, okay, it didn't translate. That's great. I don't have to explain the slides then. But really, it's about when pathways interact. We're really asking, do genes in separate pathways talk to each other in any way? Well, obviously they do, but the question is, if we take that and look across lots and lots of samples, we can ask that question. And the result is a mess. What it means, though, is we can look at different sets of pathways in the cell, and we can say, you know what? There are genes that interact with each other much more in terms of RNA metabolism than genes which are interacting between protein trafficking and RNA metabolism. We can build subnetworks of function, and that allows us to ask about things like axonogenesis, the generation of neurons. If we're doing that, what are the processes involved? We don't need to know the processes involved. All we need is a signature of this, and we can discover that. And we can do that by virtue of the fact that we can predict the interaction. So That allows us then to look at toxicogenomics databases, response to drug databases, interaction between gene set databases, and just join all the dots. This is getting fun. This is big scale stuff. Does it work? Is it real? Not sure yet. But we're doing experiments which are very promising, as they always say when you're writing a grant, and we seem to be doing all right. So we take disease signatures, we put them between a human and mouse, we look at that signature in response to these drugs, and we can identify drug clusters which might be useful. So we've done this with 100 genes that have been discovered from a survey in ALS, and we asked the question, what interacting pathways and drug responses are there? Well, what's useful is that what comes up is stuff that we'd expect, neurodevelopment, for instance. And then we've got some disease and drug response signatures that we can then go and investigate to assess the value of this methodology. This kind of method is not without its challenges. We have to understand this in terms of reproducibility, provenance, data sharing, through competitive approaches. There's a social and technical aspect to this we have to grow in terms of doing effective systems biology. We also have to have a way to handle all this data, and the cloud is the place to do that. And basically, I have this concept of cloudy with a chance of genomes. What we really have to understand is that we have to embrace technologies we don't understand very well and utilize them in this kind of space, because the scale at which this kind of data integration and interrogation is occurring is huge. So we developed a system like this at the Stem Cell Commons, which we now call the Genome Translation Commons. And what we've done is build this model. So what we do is we take information from the 100 genomes, we look at neurological disease themes, then genetic variation. We then prioritize genes we think are important using our systems model, work with companies, and end up with prognostic tools. And we've already done that. And we're very excited. I've actually been negotiating the contract this morning to go ahead and realize whether we have a predictive pathway system that tells us whether you're going to have a very bad journey in terms of motor neuron disease or not through the number of TDP, number of particles you have in your circulating through the spinal cord. So this translation commons is my vision. The action of that is occurring right now. And what we're doing is building things from machine learning and disease network models using context engines to do this. Target prioritization. Give the drug companies and give the clinicians things to go after with their drugs, tell them which drugs that might be effective and for who, and step back and watch the clinical trials happen to make more effective treatment of neurodegenerative disease. So it's happening now in Yorkshire, where I work. We have something called Health North, where we're connecting data sets together, we're recruiting and training people, we have a rollout, and effectively this is occurring day on, day in. It's very exciting. The actual delivery, I still say it's 5 to 10 years before we'll be in the clinic, but it's exciting that it's begun. So the take home is that genomics is a growing driver. Knowledge of the disease is still king. You can't do this without that. Combining specialists in knowledge with data-driven understanding is challenging, but that's what we have to do. Data scaling is an overwhelming issue, and systems and network approaches are likely to use new branches and targets. And target prioritization is the challenge. We don't really know how to select the best target. So we're getting to this concept of the data patient, which is you. We want to have predicted versus treated patients. There are trained machine learning systems in place in cancer right now at the MD Anderson Cancer System in Texas. IBM's Watson is predicting the response of patients. The relationship between genetics and environment is being helped by mobile phones, giving us information about your environment and this concept that I mentioned about you registering for the meeting, we'll be able to predict much better who gets along well with who through understanding of the genome, and that's occurring as well, and that's kind of scary. So, to finish off, we run a master's in genomic medicine at the university, it's one of 10 in the UK, and this is my team. I'd like to highlight Jerez Juárez from Mexico. He's my best student. I'm very proud of him. He went to technical college in California, and then he joined my lab at Harvard. He's the only student that joined that group at Biostats that didn't come from a well-established university in the United States. And I'm very proud of the work he's done, which I've been showing you today. The other person who's done a lot of this work is Gabriel, who invented the system called Pathwin. Muchas gracias.
sonido. So, thank you at all to our eminent panelist for his marvelous talk. Now we have almost 10 minutes for questions and answers. So remember, if anybody has an answer, just raise your hand and I will make the mic reach you. So does everybody? Okay.
Thank you, Dr. Wynn. As I told you yesterday, I continue thinking about the big challenge, particularly here in Colombia, about getting all that samples, yeah, and to get involved people that support medical research. So, can you please give us some advice about it to advance?
So let me understand your question first. Are you asking how can we access large-scale omic and genome samples in Colombia?
Oh, yeah. We have difficulties trying to get people together, try to get all those samples to make research significant.
Okay.
So it's a big issue here in Colombia. Yeah.
It takes one person. It just takes one person who understands the value. It's the same as Bill Gates, but not as crazy. You have to be an evangelist. You have to show one tangible example. And you have to say, this is what we can do. No, we can't do that. That's America, that's Boston. We can't do that here. We can do that here. I have this colleague who works at Illumina who happens to be Colombian. He or she has agreed that we can get this. I have this relationship with the World Health Organization. You build. This is what we did in South Africa. It was a very long journey. But what was exciting is that we found one victim. And the victim is usually a politician. A politician who wants to make their name as being brave but good in helping science. And this is the model that they followed in Brazil in the Xylella sequencing project. And Andrew Simpson, whose brother is a truck driver in UK, went to Brazil, married a fantastic Brazilian, and together they persuaded the Instituto Luigi to take on this project, and they did high-scale sequencing funded by Instituto Luigi by a percentage of the research budget for Brazil and by, actually, I think UNESCO. It was an amazing project. It cost a lot of money, but it had a political element to it. Now, you don't have to be brave to do this stuff. All you need are 5 samples that are from Bogotá or from Colombia that are part of a project that you submit to internationally that you just exploit. And that requires what we're doing here today, and that is building a network of powerful relationships among scientists who care and exploiting the potential that we have here in Colombia and other countries in the world to join together in international projects. That is possible, but it requires an understanding and a willingness. So the strategy that we followed in South Africa was, first of all, to attract world-class scientists who wanted to spend some time of their year in our country, and to train students and bring them back and forward between labs internationally. And secondly, to join international consortia with correct safeguards so that we could turn around to our politicians and health scientists and say, this is what we could do if we could exploit this just a little bit further. So it's a hybrid of local funding and interest and international desire to access Colombian genetic heterogeneity. And to do that effectively, you have to have safeguards so you do not lose your genetic information.
Thank you.
Renata.
So does anybody else has a question?
Dr. Hyde, perhaps one of the greatest deceptions that came out from the genome sequencing project that started in the 1980s was that realize that identifying, classifying, and annotating all the 22,000 genes that seems that we have in our genome, that classification really didn't give us clues about diseases and biological processes. So we have the list. We have a big catalog. So then came this approach that you just showed us, that how to integrate all these genes into pathways and try to realize how they interact with each other, and try to guess and bring information out from that relationships between. But when one starts to working on this field and try to work with metabolic networks or networks in general, one starts to face another problem. So my view is that we're switching or shifting the problems we're facing, because when we start to looking at the networks, we have lots of way more complicated analysis, and we need more tools in this. And when we start to look at the relationship between a couple of whatever the number of genes in one disease, you can also see that genes also participate in, or can be involved in another disease, or can be involved in another, I don't know, process where it has exactly the opposite effect that you had in the other one. Mm-hmm. So we're facing it's like a kind of exciting, but confusing time that we're living in. And what we've noticed, at least in our group, is that when you start to see, for instance, you have different layers of resolution. So if you work at the co-expression network level, you have one answer. But if you go down or go up, where you see the protein-protein interaction network, the genes that are implied in the upper layer are not the same, or the effect of this of the other layer, the protein-protein network, or you go to the metabolomics layer. So this is just to give a context of my question, which is.
You have a question?
Yeah, I have a question right now.
Yes, okay, I'm glad. Okay. I was so lost in your story. Gabriel, okay. It was like 100 years. Okay, that's good.
Okay. So now, for the cancer. So my question is that it's kind of confusing. So today, or to my knowledge, there's no really advance on how to integrate different layers of information, biological information, and we work in systems biology as a general field, mainly in one of the layers. So my first question is, have you any ideas or insights on how to work or how to integrate different layers of information into one body of knowledge so we can interpret diseases? And the other one is, how far do you think that we are from the really, really genome and personalized medicine? Okay, so
So your two questions are, the first is, how do we integrate all of this really complex multi-omic layer data effectively? And the second is, how close are we to real personalized medicine where you can walk in and I can say, I know you have a cold, right? So how close are we to that? So I'll answer the first question first. We all look at science in different ways, and if you look at integration between these layers, you read about it, and you admire how someone or a group has done an integration. It is bespoke, it is customized, it is not systematic. So you have identified the key challenge in systems biology, and it is the core of my work, systematic functional integration. Systematic is that word, which means row by column or whatever it means, okay? It's very difficult. So the only way we can address that is to establish standards, to work in larger and larger collaboratories, and to have common descriptions for the aspects of what we study, which really requires understanding that we are on a technology cycle. In Silicon Valley, where I was trained, you have the uptake phase, you have the early adopters, which is us, then you have the application phase, where everything is beginning to be used, then you have the establishment of standards, then you have the mass market. We have only begun, we're not on the derivative of that curve yet. So it's early. Our job is to be vocal about the need to do that and to generate repositories of information so that it can be integrated. Not to provide the integration, but to serve the information in a standardized way. That's all we can do now to answer that question. The second aspect of that is personalized medicine. It will be very much like a circus tent. A circus tent has huge poles, and you see the top of the tent, and it's very exciting. And those are the early wins, response to warfare, understanding that this person who walks in should not receive this drug or should have that drug. So a pharmacogenomics approach, which is simply capturing a genetic variation that's related to response to drug. That, I think, will be a very fast thing. Diagnostics as well. And really, the focus is on children. And right now, I think the most high profile, or the circus tent tops of that aspect, are looking at children who have undiagnosed diseases and sharing their genomic information with other centers where the same disease phenotype is seen, and then seeing if they can match mutations. That's a powerful thing. It's emotional, and it works. And that is an early adopting phase, which is occurring right now, and I think that's occurring globally. The troughs between the tent poles are the problem. But to attend the circus, it's so exciting, you ignore how low the ceiling is in many places. And for it to become a building will take years. To finish with that analogy, your first example of multi-omic, customized, very complex customer, it's a supermarket. The genome is a supermarket. What we have is the shelves, and the products on the shelves are now getting names. This is carbonic anhydrase, and you can find it in the carbonic anhydrase in the metabolism section. You walk through there and you pick it up. What does it do? Well, I don't know, but it tastes really good if I add it to strawberries. And I notice in Colombia, strawberries are in everything. Okay, they're in my salad, they're in this, they're in that, and I love that. They come with cheese. That's not normal, but the chef knows that they go together well, and that's the point. When you make that together, that's when you generate an understanding. You make a meal. And our job as scientists is to understand our ingredients well and to build good tasting meals, which we then serve up and we entertain people so they understand the basic understanding of that. So to get to the wine section will take us time, right? But we are effectively now at this point where we have the supermarket and we have a shopping list and we're testing out what tastes good when we put it together and understanding how that comes together.
Hello. So does anybody else has another question?
Thank you for your great presentation. Here in Colombia, we have a problem with a vaccine. Some girls were vaccinated with papillomavirus vaccine in a town in the coast called Carmen de Bolívar. And many girls get neurodevelopmental responses against the vaccine. So they experiment motor disruptions and some other disorders. And actually here we cannot understand what's going on. Do you think that could be an answer probably in the genome of this population or something that
So if I understand it, there's a negative response to papillomavirus
Yeah,
infection.
it's a negative response.
And that response is a tissue degradation response? What is the kind of response?
It manifests several stuff, the motor disorders, epilepsy, and several stuff. It's a huge group of girls. It's around 100 of girls of this small town.
Well, that's a very good question to ask. And frankly, that's the kind of example that is ripe to be executed as a poster child for effective intervention. Papillomavirus is, as you know, a huge cause of cancer. And it's a very bad story for many young women. So vaccination is essential. And a poor response to that, particularly in a country which has a disparate health system such as we have here and we have in South Africa, means that we really need to know what we're doing in order to treat our patients well. So the justification is there. The implementation of that actually may not be so difficult, or it might be a big challenge. The key is to having brothers and sisters, and to having sisters who are treated by the same viral treatment, some who respond and some who don't. If that is positive, in other words, if there are families which show differential response to this particular impact, then with that information in hand, it's possible to speculate that there's a genetic response to this. Then it's a straightforward project because there are two ways to solve that: gain international partners. And actually at the Harvard School of Public Health, a lot of the work we do is in Mexico because our president of the school was the former Minister of Health in Mexico. But I mean, there are ways to connect, or it can be done here. But the point is you have to establish a genetic concept. Is there a distribution that looks like it might have a genetic basis? Given that, it's a poster child for doing a genome approach to intervention. And I strongly suggest that you take those ideas forward. That's fascinating.
So thank you again, Dr. Hyde. And now we're going to have a 10 minutes break for a coffee